Welcome to our presentation about the hormone insulin and its biochemical mechanisms in the human body. Our names are Ellie
Mihoko.
And Elba.
and we're excited to talk to you about insulin. Insulin is produced by the pancreas, which is a glandular organ attached to the duodenum, the first part of the small intestine. It has two main roles. The first is to produce enzymes like trypsin, amylase, and lipase, which are secreted into the small intestine to help break down proteins, carbs, and lipids from your diet. The second role of the pancreas takes place at the islets of Langerhans, which two hormones, insulin and glucagon, are produced. Insulin is produced by beta cells, while glucagon is produced by alpha cells. Insulin is secreted when your blood sugar goes up, and glucagon is secreted when your blood sugar goes down. There are two types of diabetes mellitus, type 1 and type 2. Type 1 is autoimmune, in which immune cells mistakenly attack pancreatic beta cells, reducing or eliminating native insulin production. In type 2 diabetes, pancreas makes insulin, but insulin receptors aren't sensitive to it. When insulin binds to insulin receptors, downstream mechanisms don't happen in the cell, making the cell insulin resistant.
Symptoms of diabetes mellitus are often associated with extreme thirst, hunger, the need for urination, fatigue, systematic wasting, and sweetened urine. Long-term exposure of tissues to elevated glucose concentrations is associated with the development of macro and microvascular diseases, such as coronary heart disease, cerebrovascular disease, and the characteristic retinopathy, nephropathy, and neuropathy. Statistics for people with diabetes from 2015 to 2018 provided by the CDC showed that people with diabetes, that 89.9% of them were overweight or had obesity, 69% had high blood pressure, 44.3% had a high non-HDL cholesterol level, and 49.4% had a high A1C value, which indicates that they had a high average blood sugar level over the last 3 months. Other than being diagnosed with diabetes being life-altering, why should the rest of us care about it? Well, first off, 37.3 million people of all ages, which makes up for 11.3% of the United States population, have diabetes. Of that number, 1.9 million people have type 1 and the rest have type 2. Type 2 is increasing in prevalence in children and adolescents. Type 2 diabetes is the preventable illness with which healthy lifestyle habits, such as being physically active and eating healthy, can prevent or delay type 2 diabetes. Diabetes is the 7th leading cause of death in the United States, and it is now the most expensive chronic condition. 1 in every 4 US healthcare dollars is spent on someone with diabetes. 1 in $7 is spent directly on diabetes-related expenses. And in total, diabetes has costed the United States $327 billion in 2017.
Here's the structure of insulin. Insulin is a small protein composed of 51 amino acids and has a molecular weight of 5,802 daltons. It is made up of two heterodimeric peptide chains, which are the A chain and the B chain. The A chain is composed of 21 amino acids and has an N-terminal helix linked to an antiparallel C-terminal helix. The B chain is composed of 30 amino acids and has a central helical segment. These chains are linked together by two inter-disulfide bridges. There's also an additional intra-disulfide bridge on the A chain. Here, we can see the structure of the A chain on top and the structure of the B chain on the bottom. In solution, there are H bonds that are made between the B chain, C termini. This forms insulin dimers. Looking closely at this dimer, we can see 4 hydrogen bonds that are already forming. In presence of zinc, these dimers will associate into hexamers. Here is an X-ray of an insulin hexamer on the left, and on the right, an X-ray of 4 insulin hexamers in a crystal structure. So insulin synthesizes in large quantities in the pancreas by only beta cells. So first, via transcription and translation of DNA, the insulin gene is synthesized into insulin mRNA. Second, the insulin mRNA is translated as pre-proinsulin in the rough ER. Preproinsulin is a single-chain precursor protein of proinsulin composed of 110 amino acids. Its components include both the insulin A and B chains, the C-peptide chains, and the signal peptide. Thirdly, preinsulin is then cleaved rapidly to proinsulin, which is the precursor protein for insulin that is composed of 86 amino acids. Its components include both the A and B chains of insulin and the C-peptide chain that connects them. And finally, proinsulin is cleaved, removing the C-peptide, making mature insulin. This insulin and the separate C-peptide is then packaged in the Golgi apparatus into small amounts in the secretory granules near the cell membrane, and then they are accumulated in the cytoplasm awaiting secretion. Insulin and the C-peptide chain is secreted by exocytosis when the beta cells are stimulated in response to glucose. So after insulin is synthesized, it is ready and awaiting to be released. Insulin is primarily secreted when there is an elevated concentration of blood glucose. When the concentration of glucose in the blood is high, that glucose is transported into the beta cell through glucose transporter GLUT2 by facilitated diffusion. After the glucose enters the beta cell, that increases the cell's glucose concentration. This causes the glucose to undergo glucokinase and glycolysis, which will result in the release of many ATP molecules. When there is an increase in the ATP levels in the cell, potassium channels that are allowed to enter close, causing the cell to depolarize. As a result, the voltage control calcium channels will open, causing calcium to enter the cell and cause the insulin-containing secretory granules to undergo exocytosis, and insulin will be released. The C-peptides are also being released, but those are not important for insulin secretion. It should also be noted that calcium is also constantly being stored in the ER, and some of those will also be released to cause insulin release. The pancreas consists of 5 types of cells, which are the alpha cells, beta cells, epsilon cells, and the PP cells. I will give a quick overview of what these cell types do and their functions with insulin. Alpha cells are responsible for secreting glycagon, which work alongside other hormones to control the glucose levels. Glycagon stimulates the secretion of insulin, increasing its concentration. Beta cells are responsible for secreting insulin, which we have already reviewed. Delta cells are responsible for secreting somatostatin, used for producing neuroendocrine inhibitory effects to many systems. Somatostatin inhibits the release of insulin from the pancreatic isolates. Ellipsin cells are responsible for secreting ghrelin, used to stimulate the release of growth and appetite hormones. This cell decreases the plasma insulin, but also increases blood glucose levels. The relation between this cell type and insulin is still being debated today. And finally, the PP cells. The PP cells are responsible for secreting pancreatic polypeptides used to regulate the pancreatic secretion activity and the gastrointestinal movements. By inhibiting them, this makes the liver more sensitive to insulin.
Once insulin is released into the bloodstream, it travels to its sites of action at the insulin receptors. The insulin receptor is an integral membrane protein with 2 transmembrane domains. On the inside of the membrane is a tyrosine kinase domain, and on the outside are insulin binding sites. Insulin receptors simultaneously bind to each individual insulin molecule at 2 sites, one on the top and one on the bottom of insulin, inducing a conformational change. Although it is possible for 2 insulin molecules to bind, The insulin receptor exhibits negative cooperativity, so when one molecule of insulin binds, it reduces the affinity of the receptor to other molecules of insulin. Here's an animation of the insulin receptor. Insulin binds to the receptor, causing a conformational change that brings the two alpha subunits closer together. The intracellular subunits unwind, and then the autophosphorylate at 4-key tyrosine residues. Insulin receptor substrate, or IRS, is recruited and is phosphorylated itself, leading to a number of downstream signaling pathways. The insulin receptor substrate, along with a few other signaling molecules that are phosphorylated by the insulin receptor tyrosine kinase, lead to a number of downstream signaling cascades that perform a variety of actions in the cell. The most well-characterized of these functions is glucose uptake, which is what we will be focusing on in this presentation. But there are a number of other functions that are worth mentioning. Insulin binding promotes glycogen synthesis while inhibiting gluconeogenesis. It promotes lipogenesis while inhibiting lipolysis. It also activates protein and DNA synthesis, gene expression, amino acid uptake, and the sodium-potassium pump in neurons. It also inhibits apoptosis and autophagy, preventing cell death. We're going to focus on the mechanism for glucose uptake in skeletal muscle. After IRS is phosphorylated, PI3K binds and recruits AKT2, which is phosphorylated. This phosphorylates RAC1, which begins to form an actin formation that goes between the GLUT4 vesicle and the cell. RAB inhibits vesicle movement, and when AS160 is phosphorylated, it inhibits RAB, allowing the GLUT4 vesicle to merge with the cell wall, and glucose enters. In type 2 diabetes, this pathway is dysfunctional. Insulin-resistant cells typically experience decreased insulin receptor tyrosine kinase activity. Defects in the proximal signaling transduction pathway, including an IRS, PI3K, and AKT2, may also play a role. The cause of this decreased activity is still an area of active study, but it is often attributed to the genetic component of type 2 diabetes. In fact, a genome-wide association study found over 400 different genetic variants associated with type 2 diabetes. Researchers have found insulin resistance at skeletal insulin receptors in close relatives of patients with type 2 diabetes, including relatives who are not overweight. However, between 80 and 90% of patients with type 2 diabetes have obesity, so overnutrition plays a role in the mechanism of this disease. Nutrient stress from overnutrition can cause adipocyte death and cell inflammation, both of which recruit white blood cells to fatty tissues. These release JNK, which causes the release of inflammatory cytokines, such as TNF-alpha and IL-1 beta, which are hypothesized to inhibit insulin receptor activity and activate lipolysis. Triacylglycerols are broken down into non-esterified fatty acids and glycerol, which both enter the bloodstream. They travel to the liver, where glycerol raises the rate of gluconeogenesis. Fatty acids entering the liver and muscles lead to the accumulation of lipids within liver and muscle cells, which increase DAG and PKC theta signaling, inhibiting insulin receptors in the liver and muscle cells. All of these steps compound to raise the blood glucose level regardless of the presence of insulin. Long-term effects of high blood sugar can degrade blood vessels, leading to heart attack, stroke, and nerve damage. Proper management of diabetes is essential for long-term health.
I will now be talking about the broader impacts of insulin in history and in modern medicine. It is timely to talk about insulin as 2022 marks the 100th anniversary of a major breakthrough with the discovery and isolation of insulin in 1922 that drastically improved the lives of people with diabetes. This data was taken from diabetic patients attending the George Baker Clinic in Boston, Massachusetts from 1897 to 1945, where 1897 to 1913 in black bars marked the pre-insulin era, and 1922 to 1945 in gray bars marked the insulin era. As you can see in a black bar, before insulin was available, type 1 diabetes was a universally fatal illness, and type 2 diabetes resulted in poor children's life expectancy and poor prognosis for adults. During the 20th century, physicians endorsed fasting, calorie-restricted diets to decrease sugar and dietary starch intake, and for those who were obese to lose weight. At best, there was some improvement in glucosuria and acidosis, decreased coma, and delayed death among children with diabetes. At worst, People died from a reduction of eating to a point of malnourishment known as the starvation treatment. At the bottom pictures from left to right, I have shown here that other physicians followed bloodletting or gave opium. And there were a plethora of treatment fads that did not work, like Carr's remedy, Dr. Spawn's digestive elixir, Warner's diabetes remedy, white pine compound, and Brandreth's vegetable universal pills. Since 1838, people knew that glucose was a sugar that people with diabetes could not metabolize. It was hypothesized that pancreas secreted a substance that controlled carbohydrate metabolism. However, attempts to prepare pancreatic extracts that lowered blood glucose were unsuccessful due to impurities and toxicities. In 1921, at the University of Toronto, Frederick Banting thought to isolate pancreatic islet extracts called isletin from the pancreatic duct of dogs. Working with his colleagues, Charles Best and John Macleod, they showed that depancreased dogs developed diabetes, but when injected intravenously with the pancreatic extract, their blood glucose lowered. They then started to purify isletin for human use, and in 1922, the first injection to a 14-year-old boy resulted in mild glucose reduction, but with subsequent injections in the same year, it lowered blood glucose and glucosuria and ketonuria disappeared. For this outcome, Banting, Best, and Macleod received the Nobel Prize in 1923. While insulin prevented death, it was not a cure for diabetes, and insulin preparation required several injections daily. In the 1930s, H.C. Hagthorne, Scott, and Fisher was one of the scientists that sought to prolong insulin's duration of action. Hagthorne achieved this by adding protamine, while Scott and Fisher added zinc, resulting in protamine zinc insulin that lasted 24 to 36 hours. Today, insulin has developed to have shorter and longer durations of action, and insulin delivery devices have improved. As you have seen previously how insulin is involved in many cellular pathways and is necessary for survival, you would think that insulin being a necessary medication for millions of people with diabetes, that it would be affordable. In fact, the scientists behind the discovery of insulin strongly felt that insulin should be made widely available and accessible even for the poor. Unfortunately, there is no better example of big pharma price gouging insulin in the United States, driving people to other states for insulin, to other countries for insulin, to ration insulin, which leads to further health complications and even risking death. And it wasn't always this way. Since a competitor's patent could interfere or even end their research in Toronto, the University of Toronto Insulin Committee received the US patent for insulin and the methods of production. No insulin could be released for sale without their express approval, giving them control over who can manufacture the drug and how it would be done. The patent was sold for just $1 each, so that cheap insulin could be made available. Since the 1920s, insulin production required large amounts of cow or pig pancreases, limiting the production amount and making supply and cost vulnerable to price fluctuations based on gland availability. Insulin treatment had supply shortages, variable potency, and required large injection volumes. In 1922, the Toronto team failed to meet the demand of insulin production in their laboratories, so they began working with Eli Lilly Company and gave them exclusive rights to manufacture and distribute insulin free of charge to selected physicians and hospitals for just 1 year. After this 1 year, they were free to charge for insulin and patent any innovations on their product. In 1978, David Godel and his colleagues at Genentech developed the first recombinant DNA human insulin in E. coli. Following this innovation, Genentech and Lilly signed an agreement to commercialize rDNA insulin, and in 1982, the FDA approved the first biosynthetic human insulin, resolving the supply and price volatility. However, there are no real price controls in North America, even though North America only accounts for 7% of the world's diabetes. It accounts for 52% of global insulin sales. The cost of insulin continued to increase in the last 2 decades. The average list price of insulin increased 11% annually from 2001 to 2018, with an annual per capita insulin cost nearing $6,000. What was a $7.3 billion business in 2005 increased to a $21 billion global market by 2013. At the same time, the manufacturing costs for insulin has decreased. Today, regular human insulin cost of production is $24 per 750 kilograms, meaning estimated costs of production are about $48 to $71 per patient per year. The pharmacies involved in producing 99% of insulin in the US are Eli Lilly, Novo Nordisk, and Sanofi Aventis. And the main pharmacy benefit managers are CVS Caremark, Express Scripts, and Optum Rx. An investigation conducted in 2019 on the increasing price of insulin by the United States of the Senate Committee on Finance found that the business practices of and the competitive relationships between manufacturers and middlemen have created a vicious cycle of price increases. The insulin manufacturers match each other's price increases rather than competing to lower them when the efficacy of the drugs has not advanced. Novo Nordisk and Sanofi particularly closely monitored each other's price increases and increased their price in lockstep within hours or days of each other. The pharmacy benefit managers, PBMs, who act as middlemen for insurers and are the ones who set the price list of products, encourage price hikes, use rebates to compete for position on lists of prescriptions that are covered by a given insurance plan, and they take bigger cuts of the secret rebates and hidden fees that they negotiate. The industry is anything but a free market. Every part of the pharmaceutical supply chain is benefiting from the higher prices. If insulin continues to increase at the cost it has been in the last decade, gross insulin costs can reach between $60.7 billion, which would be $6,263 per patient, or up to $121.2 billion in total spending, which would be $12,446 per insulin patient by 2024. Insulin is no longer affordable for people without insurance, and even for people with insurance who needs to pay high copayments. So what's being done about the pricing of insulin? There was a legislation called the Prescription Drug Pricing Reduction Act of 2019 that sought to increase transparency of the rebates and drug pricing. And there was also a legislation introduced in the Senate called the Affordable Insulin Now Act, proposed to cap the monthly out-of-pocket cost of insulin for Americans at $35, starting October 1st, 2022, ending on December 31st, 2025. Opposition to the bill argued that it would not address the root problem of the price increases and that insurance companies would just pass on the cost through premiums. Neither legislation has passed through the Senate. Unfortunately, there is no satisfactory conclusion. It is a reality that people with diabetes must continue to face. To resolve the insulin pricing crisis, what must be done is that we see some form of government control over the pricing of drugs.